Hello, everyone. I'm very glad to have this opportunity to present some of our work on using MRI to map the electromagnetic field and the neurophysiological effects of tDCS. I'm Danny Wang from USC. I have nothing to disclose about this presentation. So tDCS is non-invasive, low-cost, minimum side effects, and it has shown therapeutic effects in a number of neurologic and psychiatric disorders. As a result, tDCS has seen exponential growth over the past decade, as shown by this PubMed search of publications. However, clinical translation of tDCS has been hampered by variable response across individuals, even in the same cohort. So our goal is to achieve personalized tDCS treatment with improved efficacy. Our approach is try to use neuroimaging for concurrent mapping the electromagnetic field induced by tDCS, as well as the brain's neurophysiological response to tDCS. As shown by this figure published by our review in 2019, we proposed to apply tDCS concurrently with MRI while scanning with innovative dual echo EPI sequence while manipulating the current intensity of tDCS. By doing this, we can concurrently acquire the magnitude and the phase. From the phase, we can derive the magnetic field change around BZ. We call it delta BZ. From the magnitude, we can derive the bold signal. And if we apply magnetic preparation, for example, spin labeling, we can acquire cerebral blood flow. So we can get functional connectivity and neurophysiological changes. How do we map the magnetic field? That's based on Ampere's law. It was first published by our group in 2016. So the curve of the B field is proportional to the current density and the time variation of the electric field. Because we apply tDCS, so this term can be crossed out. And the magnetic field change is actually, magnetic field change caused by tDCS is actually proportional to the current density around an orthogonal direction. So therefore, the magnetic field change can serve as a surrogate of the current density. So during the experiment, we would manipulate the current current intensity of tDCS during the scan while acquiring the phase maps. And we can try to estimate the phase change that is correlated to the applied current by doing a general linear model regression analysis similar to fMRI. And then we can include covariance such as head motion and the field drift, just like an fMRI experiment. So this is a phantom experiment. We have a bottle filled with water, and we apply a current carrying Y through the bottle. And then this is the BZ field. And then just by applying the Fleming's right-hand rule, you will see intuitively imagine that on the right hand, the magnetic field will be increased, while on the left side, because it's opposing the main magnetic field, it would reduce. By simulation, we demonstrate it is exactly as what we predicted. On the right side, there's increasing magnetic field. On the left side, there's decreasing magnetic field. And this is actually experimentally mapped magnetic field change. It matches almost perfectly with simulation. To further validate this technique, we changed the current polarity. So the magnetic field remain the same, except the sign flips. And this is the sham condition. We detect almost no magnetic field change. So this time course is the phase images, you see it's correlated with applied intensity, acquired tDCS intensity very well. When you flip the sign, then it is anti-correlated. While in the sham condition, there is no correlation. Further, we can demonstrate that, the fitted slope is 1 and the intercept is almost 0 when we compare experiment and the theoretical kind of prediction. We also, did an analysis within different range of magnetic field changes, and we can detect as small as 1 nanotesla per milliamp magnetic field change. This is in vivo results. We apply this technique in 7 healthy volunteers with test-retest. And this is on the left and the right motor cortex with conventional tDCS. And you can see there's a very nice correlation between test-retest with average correlation of 0.8. And this is simulated magnetic field change by using SimNIM. And we demonstrate a reasonable well correlation with experimental results with a correlation of 0.57. This results was published last year in Frontier in Neuroscience. So there's a caveat for this technique because the current will induce a magnetic field change, and the magnetic field change will causing susceptibility effects, causing both signal change. As demonstrated in this Fenton experiment, when very close, adjacent to the Y, you have strong bold signal change, less than 5%. The good news is when you are away from the Y, a few centimeters, then you get almost no signal change, just demonstrated by the time course here. With the bold signal, we can apply, standard IC analysis for network analysis. So this is just using our concurrent applied bold signal. We'll be able to detect the default mode, auditory, visual, as well executive networks, and with good correlation between test-retest. In addition, we detect one component under the cathode, one component under the anode. And the cathode cathode bold signal actually correlate with applied tDCS, negatively correlate with tDCS current under the both cast and the recast conditions. Another technique that used quite often in tDCS experiment is arterial spin labeling perfusion, mapping the cerebral blood flow change during and after tDCS. This paper was published by Dr. Nietzsche's group last year, demonstrating, with following 15 minutes anode M1 tDCS, there's consistent CBF increase, while following a 15-minute cathode tDCS stimulation, CBF reduced. And this has been further verified by TMS-induced neuronal excitability measured by MEG. With all these techniques, we sought out to apply this neuroimaging approach in a major psychiatric disease, major depression. There have been several large-scale tDCS trials on depression, but with mixed results. For example, this Bologna paper showed positive effects, while this Liu paper showed negative results. So in our case, we applied a randomized double-blind clinical trial using a Sotorix device, which allowed the double-blind design. And the neuroimaging will perform pre and post 12 sessions of tDCS treatment lasting 2 weeks. So we have 4 arms. We have a HD, we have a conventional, we also have 2 shams for HD and the conventional tDCS respectively. So the HD and the conventional each has 20 subjects, while the 2 shams each have 10. This paper was published, just published this year in Translational Psychiatry. We applied HD by the 4x1 design and the conventional with electrode with 5 by 7 centimeter. And they are all targeting left dorsolateral prefrontal cortex with a skull projection of the coordinates that is reported by the meta-analysis of TMS. This targeting and the electrode positioning was verified by brainside neural navigation during visit 6. And for neuroimaging, we applied the 3D Picasso for cerebral blood flow measurement, as well as dual echo EPI for concurrent measurement of magnetic field and both signal change. So here is the magnetic field mapping results. So this is the HD induced focal magnetic field changes in the prefrontal cortex. And under the electrode, you will see a flip from the positive magnetic field to negative magnetic field. And you can intuitively imagine there's a current injection around this line. For conventional tDCS, the induced magnetic field change are more diffuse with the prefrontal cortex. And the sham, there are very little magnetic field change. And indeed, with post-hoc analysis, we'll see, both HD and the conventional induce significant magnetic field in BA46 and 9. And we also observed a cluster on the dorsolateral prefrontal cortex on the left side for both signal change that was correlated with tDCS current. Here is the CBF results, combining the active stimulation versus the sham, pre and post the 12 session of tDCS treatment. We observed the two clusters. One is the left dorsolateral prefrontal cortex, and another one is the ACC, which are very nice, compatible with our hypothesis, since these two areas are involved in the regulation of depression. However, HD actually increased the CBF in both these two clusters, while the conventional tDCS only increased CBF in the prefrontal cortex, not in the ACC. And if we compare HD versus conventional, we'll see it actually increased CBF in the subgenuine ACC. In terms of behavior results, it's a small trial, so we didn't observe significant change in Hamilton depression scale, but in this SHAPS scale, which is indication of anhedonia, we see significant reduction of SHAPS by using HD compared to the sham, while the conventional tDCS didn't induce significant change. And this, using the bold signal, we also saw with the HD tDCS, the bold signal change also is correlated with the percentage change of SHAPS signal. So the conclusion, we saw, both HD and conventional tDCS can engage the target of the left dorsolateral prefrontal cortex by using magnetic field mapping. However, significant CBF increase in the ACC was only induced by HD, while both can increase CBF in the left dorsolateral prefrontal cortex. Further, HD significantly improved the SHAPS score, although there's no change in Hamilton score. So our preliminary data seems to suggest, we're combining both neuroimaging approach, that HD is more effective. So there's an ongoing larger scale study to further evaluate HD tDCS in major depression. I would like to thank my colleagues at USC and UCLA, as well as the CUNY group with help with the modeling, as well as funding support from NIH. Thank you.